Good morning. Thank you very much, all who have come, and in particular to the Assembly members who have sponsored this event, and to the ministry colleagues who've taken time to participate and to join us today. I've been invited to give a bit of a provocative global overview of the progress, but also the barriers that we are seeing towards non-animal approaches, not just in testing, but in health research as well. Most of us, been hearing about the 3 R's of ethical science to reduce, refine, and replace the use of animals. And we've been hearing about this for 60 years. The concept originated in 1959. So one would assume, after 6 decades of implementing 2 of the R's are to reduce or replace, if we look at statistics, if we look at the trends around animal use in science, you would expect the numbers to be going down, right? This is statistics from Canada, my home country, but you could compare these to the European Union, the United States, Australia, and here in Korea, where despite 6 decades of everybody saying they support the 3 R's, the trend line is in fact going up, and it's going up fairly dramatically. So we have a problem. Despite all of the investments that are being made in alternative approaches, very strong science, very strong commitments by industry, by regulatory authorities, the reality is that we're seeing the line going in the wrong direction. So why is that? Part of the challenge, part of the reality, is that regulatory testing is about 10% of animals in laboratories. The other 90% is basic biology, biomedical research carried out by universities and funded by research agencies, by foundations, rather than driven by regulatory requirements. So when we talk about replacement in testing, we have a list of tests, a limited number of methods as targets for replacement. When we look at what's happening in university science labs, every experiment is supposed to be original. So we don't have the potential for an off-the-shelf, one-to-one replacement the way we do in testing. So we have a different challenge that needs to be addressed. And I think, as we can see from statistics, we're not achieving that at this time. So beginning with a quote from an addiction foundation, Narcotics Anonymous, "Insanity is repeating the same mistakes and expecting different results." And while the 3 R's are not a bad idea, I think if we look at the metrics, the actual numbers, they're not sufficient for really addressing the problem. We need some fresh thinking. So I'm here to present some ideas in the context of the legislation that's going to be discussed here. And a provocative question: is it time to retire the 3 R's? Is it time for a change in paradigm? And if we compare animals in laboratories to another welfare issue, the original animal welfare movement in the West originated with carriage horses, where at the time, to travel from one point to another, horses were the most available technology, and the treatment of these animals became a subject of great public concern. And the welfare movement was created looking at how do we improve the treatment, how do we improve standards, and 150 years of efforts in that area have done very little, in fact, to address the welfare of horses. The number one thing that has actually helped these animals was the invention of the automobile. Completely different thinking, completely different innovation, but the reality is having a superior tool is something that we as human beings embrace in our lives. We like new, better, faster, more efficient, and as a consequence, the carriage horse welfare problem was solved in most of the world due to a different scientific innovation. So if we take this thinking a step further, again, Canada is a very large country. We could take our car, if we wanted to go from the East Coast to the West, and drive 45 hours, or we can use the new technology we have and take a plane. And I think all of us, despite how we may feel about the customer service we get on airplanes today, we're still going to take the most efficient option available. So if we take this thinking back into the laboratory, why, when we're talking about product safety, talking about human health, cures for diseases, are we still dealing with methods that were developed, in some cases, half a century or nearly 100 years ago? This is one animal experiment that's used for testing treatments for depression. So if we know that depression is an epidemic in many of our societies, universities and pharmaceutical companies, if they want to test a drug to treat human depression, are going to force mice to swim until they nearly drown or hang them upside down by their tails, and animals who give up, who stop trying to escape this treatment, are said to be depressed. I think if any of us know any human being suffering from depression, this is not what it looks like. This is not a valid, a predictive model for our species, but this is one of countless examples of what we call animal models that are used in the research world. As you might expect, this is not necessarily predicting what happens to you or I, and if we look at this in a grand scheme with global statistics, we're seeing that despite increasing investment in pharmaceutical development, the actual returns are going down. So the trend lines are going in opposite directions. And the consequence, when we look at medical research, is it's to benefit us. It's to help cure disease, to help create treatments. And some statistics are that 9 or 95% of every drug that's invested in by pharmaceutical companies that appears to be safe and effective in animal models turn out to be unsafe and or ineffective in people. This is a costly waste of resources. In US dollars, it's estimated to be around 2.5 billion for every new drug, taking 13 and a half years on average to come to market, and thousands of animals, for most of them to end up failing. And according to industry experts themselves, most of this failure is due to the limited predictive value of preclinical or animal tests. At Humane Society International, we have been questioning how the impact of this in different disease areas, and we've been providing grants to scientists who are dedicating their careers to tackling Alzheimer's disease, diabetes, and many other conditions. And we found through these independent studies, again and again, no disease is exempt from the fact that animal models aren't working. We've carried out a series of international workshops where we bring together these scientists with funding agencies, with regulatory authorities, and talk about what's going on, what isn't working, and how do we fix it. So we're beginning this conversation now through this assembly forum in South Korea as a very important research and innovation market. But one takeaway message that really struck me is this quote, not from an animal welfare person, but from the former director of the largest medical research funding agency in the world, the NIH. We've moved away from studying human disease in humans. The problem is that it hasn't worked, and it's time we stopped dancing around the problem. We need to refocus and adopt new methodologies for use in humans to understand disease biology in humans. So what are the new tools? What could we be doing instead of using mice and dogs and monkeys? After me, Professor Ha will give a very detailed overview of organs on a chip, so I won't say very much about this technology other than it is the future. And beyond being able to test or develop a single organ on a single chip, like the lung, the brain, the liver, the cutting edge today is to take each of these key organs and connect it with blood vessels, with oxygen, with waste removal onto a single chip. So modeling the entire human body on a small microchip-sized device. But beyond that, we have challenges in the regulatory world with hundreds of thousands of chemicals in the environment today, and the fact that we're exposed to these not individually, but as mixtures. So looking at the previous presentations from Kokvam and from Dr. Casey, the cost to test in animals for a single chemical is very high. The cost to test chemical mixtures is unaffordable. It would take hundreds of years. But with these high throughput robots at the NIH, these are able to test 1,536 chemicals in a single plate, and these robots don't sleep. They don't take lunch breaks. They work around the clock. So the throughput of these robotic high throughput cell-based testing approaches is extraordinary. Our ability to generate data through these molecular tools is unprecedented in our history. But the challenge then becomes, how do we take molecular and cellular information and apply that to a single human being, to all of us in this room? How do we connect from a mechanistic to a whole body level? We have a tremendous wealth of information and expertise within the science community. We have molecular toxicologists and biologists. We have clinicians, epidemiologists who study human populations. There's a wealth of information in the academic world today in Korea and other countries and in the published literature. The problem is it's difficult to access, and it's difficult to put this information together into one place. But that is the goal, and it's one of the objectives of the OECD today, which is managing a global development program to encourage its member countries to take their expertise and assemble it into one place. Because this is how complicated biology is. It's beyond what individual human brains can deal with. We need to leverage computer power, and we need all of these disciplines working together. So I'm going to end briefly talking about, that's the solution. What's the problem? What are the barriers? What's driving continued animal use? And the reality for the regulatory world is you have government requirements. You have the list of tests that have to be addressed to bring a pesticide, a chemical, any other regulated product to market. And the challenge is when every country has its own regulations, getting something taken out or replaced, it's not enough to do it in Korea. We need Japan, the United States, we need everyone to work together and to agree. As one example, we've been working for nearly 20 years to eliminate a one-year dog test for pesticides, and it's taken 20 years from the original research to having the final country, in this case Brazil, remove the test. We can't do this if we have 40 or 50 animal tests, 20 years apiece, to scratch them off or replace them. It's too slow. We need overarching legislation to ensure that every agency responsible recognizes and embraces this as a responsibility. And one of the models that came from the European Union is mandatory alternatives laws, to say it is a legal requirement to use alternative approaches where they exist. We have recently, in 2017, had this measure introduced into Korean chemical legislation. We're working on something similar for biocides. And again, we'll hear today about important legislation that will help to influence the wider biomedical world as well. And ending on biomedicine, here it's a different driver. It is very much driven by funding. If an agency, Ministry of Health, Ministry of Science, provide funding, how they characterize that opportunity matters tremendously. If there's 100 billion won, for instance, for cancer research, lung cancer research, and the agency sets no limitations, we're going to see a lot of animal research probably. But if they said, we would like to see lung cancer on a chip using human cells as the model, suddenly that's going to drive the scientists who provide the funding proposals in a very different direction. And on the one hand, we've seen the numbers go up when animal research if the mouse is considered the starting point in a conversation, that's where the conversation will probably end. But if a decision is made from the very beginning, from those who control the funding, that a human-based approach is what's desired because we're trying to predict human biology, not mice, not dogs, that makes a difference. We're beginning to see this translating. This is a slide from the European Commission where they've acknowledged there are many animal models that are poorly predictive. And a key policy in Europe is now replacing animals with better in vitro, cell-based, and in silico computational models. In the United States, we're seeing legislation beginning to be introduced that is, again, creating a framework. In this case, it's called the HARTS Act, Humane and Existing Alternatives in Research and Testing Sciences, which is somewhat similar in scope to the legislation that's being proposed here. So to wrap it all up, the two things that we recognize really need to be addressed are, for testing in the regulatory area, mandatory alternatives as a legal framework to ensure harmonization across the globe, and a redirection of research funding in the biomedical and basic sciences, which will address the 90% of animal use in laboratories. So with that, I'd like to thank you all for your attention and very much looking forward to investing in the future. Thank you.